For years, I've been working on social organisms or social arthropods that are not ants or bees or wasps or termites. So these are sometimes called the other social insects, and they are sometimes not well-known social insects or insects that live in groups and express more than just group behavior, but some sort of cohesive social behaviors that reminds us of the social behaviors that we see in the social Hymenoptera and the termites. So my research for years has been, or what I talked about at ESA, was trying to think about or catalog patterns of division of labor. So some individuals who reproduce, some who do the work in a group, in things that may not be familiar to a lot of entomologists. If you choose to study a non-model organism for which there's not a lot of backstory, there's not a lot of tools, there's not a lot of history, there's not a lot of work that people have done for ages, you're having to create some of the fundamental tools that you need to study that organism from scratch. These are non-model organisms. And one of the reasons you discover very quickly why people have not studied them is because it's hard to study them. And so there's a reason for that. So there's a reason that Morgan chose Drosophila, as his model organism, is because it had practical features that made it a good model organism. And so when you choose a non-model organism, like social aphids are by definition, you are running up against practical challenges like culturing them in the lab or having the tools to actually ask the kinds of questions that you want to ask. There's been a century or at least of work on the chemosensory biology of an aphid. So aphids are pests of our plants. We don't like aphids. They threaten our food security. They vector viruses. They cost us literally over the decades billions of dollars in terms of crop losses. And we put a lot of energy and time into managing their populations and their outbreaks and preventing them from being on our plants. So that means there is a reservoir of information about chemosensory biology, for example, of aphids in general, that can be leveraged to study social aphids. So for example, at the level of chemosensory or input or olfaction, how does a social or soldier perceive another insect, a predator, a threat, right? And so what, for example, are the chemosensory signals that by which soldiers actually recognize a threat? So the directions that my research would like to take, or is hoping to take, is looking more mechanistically and functionally at what makes a social aphid. So that's what I'm doing next.